Viruses, if we back up for a moment, have been largely miscategorized as part of the microbiome. The microbiome, composed of two obvious words. The first, micro, meaning small. The second, meaning biome, which means living organism. And it turns out that viruses do not fall into that category of microbiome because they're not actually living organisms. They do not produce energy. They can't reproduce. They're literally a package of genetic information. They don't have any enzymes that can reproduce that DNA within them or anything else. So they've really a communication network of the genome. And so viruses as a category have been miscategorized into the microbiome for many years. And in fact, if you read viruses, definition of the microbiome, it'll often say include viruses. But if you then ask why viruses, it'll say, well, because they're really, really small. And so they drop the second word biome and say, but since they're super, super small, we should just call them microbiome anyways. That seems like a relatively small mistake to make, unless it turns out that you then take all of your experiential, germ theory that you've developed around bacteria and gangrene as we started understanding it in the Civil War and everything else, and then apply that to a non-living life form like a virus. It turns out we misconstrue the entire matrix of the virome. The virome is its own ecosystem of genomic information that's emerging from bacteria and multicellular life. At least 50%, and I won't be surprised if it's 95% in the end, but at least 50% of the genomic information that we call viruses is being exuded by bacteria. And those we call bacteriophage. And so instead of calling those viruses, we'll typically call them bacteriophage, but bacteriophage is the translation and transportation of genetic information from one bacteria to a next, and then it goes inside. If the bacteria chooses to reproduce that, it will start manufacturing that and redistribute that back out into nature in a higher volume. So it can amplify that genetic signal. Interestingly, whether it be human or a bacteria, the cell has a lot of regulatory functions as to decide which DNA or RNA is allowed to enter into the cytoplasm of the cell and which DNA or RNA is allowed to survive long enough to interact with the highly regulated machinery that will determine whether you will actually make a protein and allow that gene to turn on or not. And so this is a highly regulated communication network of genomic updates. And viruses are produced with the very mission of creating adaptation. A virus will never, go out into nature and do the same thing that another genome already does all the time. It's not necessary. And so it doesn't waste the effort of repeating itself if that genetic information has already been brought into the physiology of an organism and is now in a homeostasis or balance with that genetic experience of being a live organism. For this reason, we don't continue to produce the same virus over and over again. We will take a virus in, we'll produce it for a short period of time. We take that into the tissues of our body that most need that genetic information, and then we reach a balanced state. If you get influenza, a specific species or strain rather of that genetic code, you will update that whole genomic sequence within you. Interestingly, we now know that the DNA is constantly updating. Some of this DNA will hold on to that information. The herpes viruses and some of these chronic viral conditions out there mean that genome has been supplanted into your long-term storage devices of your genome. The human genome, as now understood through sequencing, has been composed of over 50% of its volume from viral inputs. 10% of the genome, in fact, has been inserted directly by retroviruses like HIV. And so the genome that makes us human today is a billions of years journey into the construction of the complexity and intelligence of this genomic information inserted into us by viruses. I want to emphasize how important these viral genomic things are. These aren't like, oh, I got exposed to a germ and I accidentally integrated that into my system and now I keep it at bay. No, these are important, critical updates to our adaptive capacity as human beings. Good examples of this are, in fact, the genome of stem cells. And so a stem cell is actually not able to be pluripotent, meaning it can change into a liver cell or a bone cell or a kidney cell, unless it has the genetic update of a retrovirus that was inserted millions of years ago. And so through a retrovirus, we gained the function of a pluripotent stem cell as multicellular organisms. Same thing actually gets even more interesting in that the mechanism by which we produce the placenta, to have produced the very first human being, the placenta needed retroviral and viral information genes that regulate that production of the placenta. So if not for viral updates to our genome, we would have never had the adaptive capacity and the biologic intelligence to birth a single human being. So how is it that we have so screwed up with this COVID thing, a demonization of the very genetic code that has produced life on Earth? But unfortunately, that's what we've done. And we've done it really with an ulterior motive. And it's been interesting to see for the first time the pharmaceutical industry and the mechanisms of the WHO and CDC really show their cards for the first time, and that their entire agenda seems to be targeted at the production of a multi-billion-dollar vaccine. They didn't seem to have the concern of, showing us how we could be in a balanced state with this virus, which is, of course, happened every other time we've seen a coronavirus. We saw SARS and we saw MERS, and within a single year, within 2 years, or 2 seasons, so a single 12-month period encompassing 2 of our respiratory seasons, SARS disappeared, no vaccine. 2012, MERS disappears, no vaccine. We didn't need a vaccine because the genetic update had occurred. We developed a homeostasis or balance within the populations that had been exposed, figured out how to get that update in, and we're no longer producing the virus. They didn't do that by killing the virus. We have this belief that we need antibodies to kill the viruses so that we're not, you don't kill a virus, it's already dead. You don't need to kill the virus. What you need to do is keep the machinery intact and communicating well, that then regulates the generation or silencing of that genetic information as it enters the body. And so we have demonized the virome to our own demise because we've made public health decisions that have isolated us rather than reconnected us to nature. We have made public health decisions that have collapsed global economies, lost 250 million jobs over the last 4 months. We have destroyed the international community on many levels. And of course, the most poor, the most marginalized, the most abused, minorities among us were the worst hit by these mistakes. And so it's sobering to realize that the misrepresentation of the virome as part of the microbiome, and therefore the attribution of germ theory upon these dead, non-living genetic, updates, we've made some critical mistakes scientifically, medically, and policy-wise.